Life on Earth might be far stranger than we ever believed. Hidden in plain sight, a secret world could exist right under our feet, a realm scientists call the Shadow Biosphere. This is the mind-bending idea of a hidden ecosystem, a form of life with a completely different biological foundation than anything we have ever known. We will journey through the evidence and possibilities, and by the end, you will understand how finding this second genesis of life would rewrite everything we know about biology. We will investigate how such life could remain hidden for so long and what it would mean if we are not alone on our own planet. Does the idea of an entirely separate tree of life sharing our world spark your curiosity? A shadow biosphere is not merely a collection of undiscovered creatures. It is something far more profound, a complete biological system operating on a different set of rules. Think of all life as we know it, from the smallest bacterium to the largest whale, as being built from the same fundamental blueprint. We all use DNA for our genetic code, RNA as a messenger, and proteins built from the same 20 amino acids. This shared heritage points to a single origin, one common ancestor from which every living thing on Earth descended. A shadow biosphere shatters that unity. It represents a second origin, a separate genesis for life that would run on an entirely different operating system. If known life is a personal computer, a shadow biosphere would be an Apple computer. Both are complex machines that process information, but their fundamental architecture, their very language of operation, is incompatible. You cannot simply take a program written for one and run it on the other. In the same way, weird life might not use DNA at all. Its genetic information could be stored in a completely different molecule. It might build its cellular machinery from amino acids that are mirror images of our own, or from a set we have never encountered. Its metabolism could draw energy from chemical sources we consider inert, or thrive in environments we deem utterly sterile. This is why the idea is so revolutionary. Finding a single microbe with an alternative biochemistry would be the most monumental discovery in biology since Darwin. It would prove that the spark of life is not a freak, one-time accident, but a more common and perhaps inevitable cosmic event. Our planet would not just host life, but lives, a hidden neighbor sharing our world while remaining utterly alien. Our entire known world of biology rests upon a deeply ingrained set of rules. At the heart of every cell in every plant, animal, and fungus is a universal instruction manual written in the language of DNA. This genetic code is transcribed into a working copy called RNA, which then directs the assembly of proteins, the complex machines that carry out the work of life. This elegant flow of information from DNA to RNA to protein is so fundamental it is called the central dogma of biology. It is the operating system for life as we know it. But the story does not end there. The proteins themselves are built from a very specific set of just 20 amino acids, a standard toolkit used by every organism we have ever studied. These proteins operate within cells that are all enclosed by a fatty, protective barrier called a lipid bilayer. And all of this intricate molecular dance happens suspended in a universal solvent, the one substance we cannot seem to live without: water. For generations, scientists have reasoned that these are not just quirks of our particular branch of life. They appeared to be universal requirements, the only way to build a stable, self-replicating system complex enough to be called alive. This created a powerful assumption, a biological blind spot. We have been searching for life by looking for reflections of ourselves. Our microscopes, our chemical tests, our very definition of what is alive are all calibrated to detect this specific carbon-based, water-centric biology. We are like people who have only ever seen oak trees, so we walk through a forest looking only for acorns, completely unaware of the pine cones scattered at our feet. This deeply ingrained perspective is the very reason a shadow biosphere could exist, hidden in plain sight. If an alternative form of life used a different genetic molecule or a mirror image version of our amino acids, our most sophisticated laboratory tests might simply register it as background noise, as non-living chemistry. We would be blind to it, not because it is not there, but because we have never known what to look for. The idea that we might be sharing our planet with an entirely alien form of biology did not simply appear out of thin air. It was carefully constructed by a handful of visionary scientists who dared to challenge the deepest assumptions of their field. Among the most prominent voices are the physicist Paul Davies and the philosopher of science Carol Cleland. In the early 2000s, they began to articulate a powerful and provocative question. If life arose more than once on Earth, where would we find the evidence? They argued that our planet is the only place in the universe so far where we know for certain that life exists. Therefore, it is the most logical place to search for a second genesis. Their reasoning was a direct challenge to the biological status quo. For decades, the search for extraterrestrial life focused on distant planets, while the possibility of a hidden biosphere right here was largely ignored. Davies and Cleland pointed out that our methods for detecting microbial life are incredibly biased. We only find what we are already looking for. We culture microbes in our labs using nutrients that suit our kind of life, and we sequence genetic material that matches our own. Any life form built from different chemistry would be completely invisible to these tests. It would be like trying to tune a radio to an FM station while the signal we are searching for is broadcasting on an AM. This line of thinking began to shift the shadow biosphere concept from the realm of science fiction into a legitimate, though still highly speculative, scientific hypothesis. The groundwork for this radical openness, however, was laid decades earlier with a series of stunning discoveries on our own world. Throughout the 1970s and 80s, biologists began finding life in places once thought to be utterly sterile and inhospitable. They discovered extremophiles, organisms thriving in boiling hot acid, in the crushing pressures of the deep ocean, and in rocks miles beneath the Earth's surface. These resilient creatures were still our relatives, still using DNA and the same 20 amino acids. But their existence proved that life could persist in environments far stranger than we had ever imagined. If our own kind of life could flourish in such extreme conditions, then why could not a completely different form of life be hiding in those very same niches or in places even more exotic? The discovery of extremophiles blew the door wide open, forcing scientists to confront a humbling truth. We had vastly underestimated the tenacity and versatility of life. The search was no longer about finding life where we expected it, but about learning how to see it where we did not. The shadow biosphere hypothesis was thrust into the global spotlight in 2010, not with a whisper, but with a scientific declaration that shook the foundations of biology. NASA held a press conference to announce a breathtaking discovery, one that seemed to leap straight from the pages of a science fiction novel. A team of researchers claimed to have found a bacterium, strain GFA-J1, living in the toxic, arsenic-rich waters of Mono Lake in California. This was not just another extremophile tolerating a harsh environment. The scientists asserted that this microbe was doing something previously thought impossible. It was incorporating arsenic, a poisonous element, directly into its DNA, using it in place of the essential element phosphorus. The implications were staggering. If true, this was not merely a new species, it was a form of life with a fundamentally different biochemical backbone. Phosphorus is a cornerstone of all known life, a crucial part of the structural framework of DNA and the energy currency of the cell. To replace it with arsenic would be like building a skyscraper with a different, unstable metal and having it stand tall. This would be the first concrete evidence of that second genesis, a life form playing by a completely different set of rules, living right here on Earth. The initial wave of excitement was immense, promising to rewrite biology textbooks overnight. But in science, extraordinary claims demand extraordinary evidence, and the scrutiny was swift and severe. Within weeks, a chorus of criticism arose from the broader scientific community. Other biologists began to dissect the NASA study, pointing out what they saw as critical flaws in the methodology. The primary critique was devastatingly simple. The critics argued that the bacteria were not actually using arsenic to build their vital molecules. They were merely surviving in its presence, a feat many extremophiles are known for. They suggested that the traces of arsenic found in the bacteria's DNA were not structural replacements for phosphorus, but merely contaminants clinging to the molecules. The bacteria, they said, were likely still scavenging every last precious atom of phosphorus from the contaminated growth medium, not happily building with poison. The scientific process, with its rigorous demand for proof and reproducibility, began to dismantle the initial claim. Follow-up studies by independent labs failed to replicate the original findings, and the consensus gradually shifted. The GFA-J1 bacterium was not the revolutionary herald of a shadow biosphere. It was a remarkable example of extreme tolerance, but still very much a part of our own familiar tree of life. This entire episode serves as a powerful case study in the immense challenge of proving alternative biochemistry. It highlights the fine line between a groundbreaking discovery and a compelling but flawed interpretation. The controversy was not a failure of science, but a demonstration of its core strength, its self-correcting nature driven by skepticism and verification. The search for a shadow biosphere requires us to not only imagine the unimaginable, but to prove its existence against a backdrop of our own biological noise. We are trying to hear a faint, unfamiliar melody while standing in the middle of a roaring orchestra that only plays one song. The arsenic bacteria story, for all its controversy, sharpened the tools and the questions. It forced scientists to ask with even greater precision what would constitute definitive proof of a second genesis. If not arsenic in DNA, then what are the true, unambiguous signatures of a life form built from an entirely different blueprint? So if the arsenic affair taught us anything, it is that we need to look for stranger, more fundamental differences. The true signatures of a second genesis would not be a simple element swap, but a complete rewrite of life's core architecture. One of the most compelling alternatives is the idea of silicon-based life. Carbon is the backbone of our biology for a brilliant reason. It can form 4 strong, stable bonds, allowing it to create the long, complex chains and rings that make up proteins and DNA. Silicon sits directly below carbon on the periodic table, meaning it shares this ability to form 4 bonds. In theory, it could assemble into its own complex structures. Science fiction has long dreamed of silicon creatures, slow-moving crystalline beings living over geological timescales, but the reality is far more constrained. Silicon has a critical weakness. Its bonds with oxygen are extremely strong. On our world, bathed in oxygen and water, silicon readily locks up into silicon dioxide, which we know as sand and rock. Sand is, for all intents and purposes, silicon ash, a dead end for the complex dynamic chemistry required for life. A silicon-based metabolism would likely need a very different environment, perhaps one devoid of free oxygen, where it could form more flexible molecules like silanes, analogues to our hydrocarbons. Yet even there, silicon-silicon bonds are weaker than carbon-carbon bonds, making long chains less stable. This does not rule silicon life out, but it suggests that if it exists, it would be a quieter, more fragile biology, perhaps hidden deep within anoxic rocks, completely isolated from our vibrant, carbon-based world. The very solvent of life is another pillar we take for granted. Water is miraculous, a universal solvent that can dissolve nutrients, transport molecules, and maintain the delicate structures of our cells. But what if life used a different liquid, Imagine a world where the oceans are not water, but liquid methane, with temperatures dipping to negative 180 degrees Celsius. This is not a fantasy, it is Saturn's moon Titan. In such bitter cold, the chemistry of life as we know it grinds to a halt. Water is a solid, unforgiving rock. Yet, in that methane sea, a different form of biology could theoretically emerge, one built on slower, colder reactions, using substances like ethane and acetylene as its energy sources. Its cells would not be water-based blobs, but perhaps structures built from lipids that remain flexible in the deep freeze. Ammonia is another candidate, a solvent that remains liquid at much lower temperatures than water and can dissolve many organic molecules. A shadow biosphere on Earth could theoretically harbor pockets of such life in environments where ammonia exists in liquid form, perhaps deep underground or in specialized chemical soups. These life forms would be so alien that our water-drenched world would be a scorching, poisonous hell to them. They would not compete with us for resources because their resources would be our poisons and ours would be theirs. Perhaps the most subtle and therefore most plausible candidate for a shadow biosphere is right in front of us, built from the same pieces but assembled in mirror image. Many of the molecules of life are chiral, meaning they come in two versions that are mirror reflections of each other, like a left and a right hand. Our biology is almost exclusively left-handed when it comes to amino acids and right-handed for sugars. This is a profound mystery, a frozen accident from the dawn of life, but there is no chemical reason why the opposite could not be true. A mirror life form would be built from right-handed amino acids and left-handed sugars. To our instruments, its molecules would appear identical in composition, but their structure would be the wrong-handedness. This has a stunning consequence. Our enzymes, which are like highly specific locks, would not fit the keys of mirror life. Its proteins would be indigestible, its DNA unreadable. A mirror life microbe could float right into our body, and our immune system would not even recognize it as life. It would be biochemically invisible. Such an organism would not need a distant, exotic habitat. It could be living in the soil of your garden, in the same puddle as ordinary bacteria, completely unnoticed because our tests are blind to its handedness. It would be a ghost in the machine of our own biosphere, a separate creation forever passing through our own without a single interaction. Each of these possibilities, from silicon foundations to alien solvents to mirror images, paints a picture of a potential hidden Earth. They are not competing ideas, but different pathways a second genesis could have taken. Finding any one of them would mean our planet is not just a home for life, but a cosmic library containing at least two entirely different volumes of the Book of Life. The most promising hiding places for a shadow biosphere are not the lush forests or teeming oceans, but the forgotten corners of our world, places so harsh and isolated that they represent a form of planetary exile. Deep beneath our feet lies a realm of profound darkness and pressure, a world utterly divorced from the sun's energy. Miles below the Earth's crust in the deep subsurface, temperatures soar and pressures become crushing. Here, no photon of sunlight has ever reached. For our kind of life, this is a starvation diet, but for an alternative biology, it could be a sanctuary. Our life depends on photosynthesis, either directly by eating plants or indirectly by consuming creatures that do. But in the subsurface, the only energy comes from the slow, relentless decay of radioactive rocks and the grinding heat of geology itself. Microbes from our tree of life do eke out an existence here, metabolizing hydrogen, sulfur, and iron from the surrounding rock. But what if another form of life, built to thrive on even scantier energy sources, called this place home? A shadow biosphere organism might draw sustenance from chemical reactions we consider far too weak or slow to be useful, giving it a decisive advantage in this energy-poor desert. It would not compete with our life because our life is barely there to begin with. it would have the entire deep Earth to itself, a hidden continent spanning the globe. Then there are the hydrothermal vents, the spectacular black smokers on the ocean floor. These are not peaceful gardens, but violent chimneys spewing superheated, mineral-rich water into the icy darkness. They are cauldrons of bizarre chemistry, where hydrogen sulfide and methane bubble forth from the planet's mantle. Our life has adapted to these extremes, forming lush ecosystems of giant tube worms and strange clams. Yet within these very plumes, amid the chaotic mix of chemicals, could reside a chemistry so alien it has gone unnoticed. A life form that prefers the specific, exotic compounds formed only under these extreme pressures and temperatures, a biology that treats the vent's toxic output as a gourmet feast. Moving from the deep ocean to the driest land, we find the hyper-arid deserts like the Atacama in Chile. This is a place where rainfall is measured in millimeters per decade, and the soil is so depleted of organic nutrients that it is often used as an analog for Mars. Our microbes cling to life here in a state of near-permanent hibernation, but for a shadow biosphere, this could be a refuge. If an alternative life form did not rely on water as its solvent, but on something like formamide or concentrated brine that resists evaporation, the Atacama's dryness would be no threat. It could flourish in the salt crusts or within the parched soil, completely invisible to our tests that look for water-based metabolism. Even the most mundane places could conceal profound secrets. Consider the tiny, nanoscale pores within seemingly solid rocks. To a microbe, a granite slab is not a solid object but a porous, labyrinthine city. Our bacteria can live in these tiny spaces, but they are at the very limit of what is physically possible for a water-based cell with DNA. A shadow biosphere life form, perhaps one with a simpler, more stable genetic molecule or a smaller, more robust cellular structure, could thrive in pores too small for our life to inhabit. It would be a world within the world, a miniature ecosystem hidden inside the very stones we walk upon. From the crushing depths to the high-altitude, salt-crusted lakes, these extreme niches share a common thread. They are places where the rules of our biology are stretched to the breaking point. For us, they are the edges of the habitable map. For another form of life, they could be the heartland. But the search for a shadow biosphere might not require digging into rocks or descending into ocean trenches. The evidence could already be inside us, hiding within the most enigmatic biological entities we know, viruses. Viruses occupy a strange borderland between the living and the non-living. They carry genetic information, they evolve, but they cannot reproduce on their own. They are pirates hijacking the cellular machinery of their hosts to make more copies of themselves. For the most part, the viruses we study are pirates that prey on our own tree of life. But what if there are other viruses out there, privateers sailing for a completely different fleet? This leads us to the shadow virus hypothesis. When scientists sequence the genetic material of viruses found in the environment, they often stumble upon a profound mystery. They find genes that have no known counterpart in any cellular organism on Earth. These are called orphan genes. They are stretches of genetic code that seem to come from nowhere, with no evolutionary relatives in our entire database of life. The question is haunting. Where did these genes come from? One compelling possibility is that they are genetic fragments from a shadow biosphere. They could be the discarded blueprints or the active weapons of a completely alien biology, hitchhiking on our own world. Imagine a shadow virus, one that is built from an alternative genetic molecule. It could infect a cell from our biosphere, but because its chemistry is incompatible, it might not cause the cell to burst or produce new viruses in any way we would recognize. It might simply sit there, dormant and undetectable, or it might trigger subtle, strange metabolic changes that we would dismiss as unexplained cellular noise. Our standard tests for viral infection are designed to find what we already know. They look for the genetic signatures of familiar pathogens, a shadow virus with its fundamentally different code would be invisible. It would be a ghost in the machine of our own bodies. This is where modern science is beginning to shine a light. Metagenomic studies allow researchers to sequence all the genetic material in a sample from a particular environment, like seawater or soil, without needing to culture anything in a lab. This approach has uncovered a vast universe of viral diversity, a genetic dark matter that vastly outnumbers the genes of all cellular life combined. Buried within this dark matter could be the true signatures of a second genesis. Scientists are now developing new tools to sift through this data, not looking for genes similar to known ones, but searching for sequences that are radically different, for patterns that defy the standard logic of our biology. They are hunting for genetic aliens in a sea of our own information. Finding a single virus with a completely novel biochemistry would be a monumental clue. It would suggest we are not just sharing our planet with a hidden cellular life, but that we are entangled with it, that its genetic messages are drifting through our environment and perhaps even through us, a silent, parallel existence written in a language we are only just learning to see. Finding these hidden forms of life demands a radical shift in our methods. The first and most obvious approach is to try and grow them, a process called cultivation. For centuries, microbiologists have discovered new species by taking a sample from the environment and offering it a meal in the lab, a nutrient broth designed to encourage growth. But this entire process is built on a towering assumption that the life we are searching for eats our food and breathes our air. A shadow organism would likely starve to death on our gourmet bacterial feasts or find our oxygen-rich atmosphere utterly poisonous. If it uses arsenic for energy, our standard media would be a barren wasteland. If it thrives on solid rock, a liquid broth would be a drowning pool. The very act of trying to grow it with our methods would likely kill it. So researchers are designing radically new menus. They are creating growth chambers with atmospheres of pure hydrogen sulfide or bubbling methane through icy slush to mimic the conditions of Titan. They are feeding potential shadow life a diet of pure silica or crystals of mirror-image amino acids, hoping to finally offer the right key for a lock we have never seen. This is a process of profound humility, an admission that we must let go of our assumptions about what life needs to flourish. Alongside these novel banquets, scientists are peering into the microscopic world with new eyes. Advanced microscopy allows us to see not just the shape of a cell, but its internal composition. Imagine looking at a sample from a deep-sea vent and finding a cell that lacks the standard fatty membrane, its boundary instead a crystalline shell or a gel-like matrix. Or discovering an organism that shows no internal structure we recognize, no nucleus, no ribosomes, just a swirling, alien architecture. These strange morphologies would be a powerful clue, a signal that we are not looking at a new branch on our tree, but at a tree from an entirely different forest. Dedicated projects are now underway, scouring the most extreme environments on Earth, from the high-acid rivers of Spain's Rio Tinto to the ultra-dry valleys of Antarctica. They collect samples and subject them to a battery of hundreds of different growth conditions, a massive, systematic fishing expedition in the chemical dark. They are not just looking for life as we know it, they are casting a net of unimaginable breadth, hoping to finally snag a life form that, until now, has had no reason to reveal itself. The hunt for a shadow biosphere has entered its most sophisticated phase, moving beyond microscopes and Petri dishes into the realm of pure information. This is the frontier of metagenomics, a revolutionary technique that allows scientists to sequence every single strand of genetic material in an environmental sample, whether it is from deep ocean mud, desert soil, or the air itself. Instead of trying to grow one microbe at a time, researchers can now take a literal census of an entire microscopic ecosystem, reading the genetic code of thousands of organisms simultaneously. It is like having a machine that can identify every book in a vast library just by scanning the shelves. This approach has uncovered a breathtaking diversity of life we never knew existed, revealing countless new branches on our own family tree. But the true quarry is far more elusive. What if, amid this sea of familiar genetic code, there is a book written in a language we have never seen? What if shadow life does not use DNA at all? Our entire metagenomic method is built to read the language of DNA and RNA. It is a brilliant tool, but it is utterly deaf to any other language. If an organism uses an alternative genetic polymer, a completely different molecule to store its information, it would be completely invisible to our sequencing machines. It would be a ghost in the data, a profound silence where a signal should be. This realization has spurred a new, more subtle kind of search. Scientists are no longer just looking for positive signals of life. They are actively hunting for the absence of the familiar. They are scanning their genomic data for gaps and anomalies, for pockets of an environment that seems strangely sterile. If you find a sample from a deep, hot rock formation that shows no trace of known microbial life, that void itself becomes suspicious. Why is there nothing there? Is it truly barren, or is it filled with a biology that our tools cannot register? This is where chemical forensics takes over. Researchers are developing methods to look for molecules with unusual atomic compositions. Remember the arsenic bacteria hypothesis? Even though that specific claim was disproven, the underlying idea was sound. Scientists can now analyze biological samples not just for their genetic sequence, but for their elemental makeup. They can look for cells that have a strange ratio of elements, where carbon is replaced by silicon in their structures, or where phosphorus is mysteriously absent. They are not just reading the text of life, they are analyzing the very paper and ink it is written with, searching for a page made of something other than paper. These techniques are the equivalent of building a detector for a substance we have never actually seen. We are looking for the shadow cast by an invisible object, listening for the quiet in a room we thought was empty, hoping that silence itself will finally lead us to our hidden neighbor. This vast, uncatalogued wilderness of microbes has a name: microbial dark matter. Just as astronomers look out at the universe and see that most of its mass is invisible, made of a substance they cannot directly detect, microbiologists peer into their microscopes and know that most of the tiny cells they see are complete mysteries to us. We can observe them, count them, but we cannot make them grow in our laboratories. The overwhelming majority of these uncultivated microbes are almost certainly part of our own tree of life. They are just incredibly fussy about their living conditions, requiring a specific combination of nutrients, pressure, or microbial neighbors that we have not yet been able to reproduce. But within that immense sea of unknown life lies a staggering possibility. What if a small fraction of this microbial dark matter is not our long-lost relatives but absolute strangers? What if some of those cells that stubbornly refuse to grow on our agar plates are not being difficult, they are being alien, because the very chemistry of our lab food is as useless to them as a plate of gravel would be to us? This is where the search becomes a technological detective story. Scientists are now using a powerful technique called single-cell genomics to break the cultivation deadlock. They can carefully isolate a single, mysterious cell from an environmental sample, even one that has never been grown in a lab. Then they amplify its entire genetic blueprint, reading its code from just that one specimen. This method has already pulled countless new branches on the tree of life out of the darkness, revealing bacteria and archaea we never knew existed. But the real hope is to find a cell that defies all classification, one whose genetic code is written in a script so foreign that it does not even use the same alphabet. Imagine sequencing one of these dark matter cells and finding it has no DNA. Instead, its entire instruction manual is written in a molecule we have never seen, a genetic polymer built from an entirely different set of chemical rules. That single discovery would shatter our understanding of biology forever. It would be the ultimate signal emerging from the noise, proving that we have been sharing our planet with a second genesis for eons, a secret co-inhabitant hidden within the vast and uncharted census of microbial dark matter. The tools to make this discovery are now in our hands, and with them, we are finally beginning to read the missing pages of life's story. Finding a second genesis of life on Earth would transform the mystery of our own beginnings. For centuries, the origin of life, abiogenesis, has been seen as the ultimate cosmic lottery ticket, a one-in-a-trillion series of chemical accidents that culminated in that first, fragile, self-replicating molecule. The odds seemed so astronomically slim that we appeared to be a miracle, a solitary flicker of consciousness in a vast, sterile universe. But if we discovered that life had ignited twice, independently, on this very same world, that entire narrative would collapse. It would mean the spark of life is not a desperate, improbable flame, but a fire that readily catches wherever the right conditions persist. Our planet would no longer be a statistical outlier, but a standard model. The universe, instead of being mostly empty and silent, would likely be teeming with life. We would look up at the night sky with a new and profound certainty, knowing that biology is not a freak event, but a fundamental and regular feature of cosmic evolution. The question then becomes not if life exists out there, but what forms it has taken. This discovery would force a dramatic re-evaluation of the very first chapters of life's story. Did this second genesis occur completely separately from our own, emerging from its own unique probiotic soup in a different hydrothermal vent or a separate tidal pool? Or did both our life and the shadow life branch from a shared, earlier stage, a primitive proto-life that was not yet fully committed to DNA or its alternative? This is a deep and unresolved puzzle. One theory suggests that the first living systems were far more flexible, a chaotic molecular society where many different information molecules competed. In this primordial arena, our DNA-based life simply won out, becoming the dominant biology and eventually eclipsing all other experiments. The Shadow Biosphere, then, would be a relic, a surviving remnant of that lost era of biological experimentation, a living fossil from the dawn of creation. The alternative is even more mind-bending, that two entirely separate lines of life arose from completely independent chemical pathways, perhaps even at different times in Earth's history. One genesis might have occurred in the scorching heat of a volcanic vent, while the other began in the icy films of a clay mineral. They would be true strangers, meeting for the first time after billions of years of separate evolution on the same planet. Either scenario reshapes our search for life beyond Earth. It tells us that we should not just look for planets that mirror our own environment, but for any world with a rich and varied chemistry. A second genesis on Earth proves that the universe is not just habitable, it is inevitably, gloriously alive. A second genesis on our own world would be the single greatest discovery in the history of astrobiology. It would transform the cosmic question from a whisper to a roar. The universe would no longer feel like a vast, empty museum, but a thriving, living library. If life could start twice on one planet, then it is not a fragile miracle, it is a cosmic imperative. The silence of the cosmos would not be a sign of emptiness, but a sign that we have been listening for the wrong things. Our current search for life is built entirely on a sample size of one. Every instrument we have ever sent to another world, from the Viking landers on Mars to the sophisticated labs on the Perseverance rover, is calibrated to detect the specific chemical fingerprints of our own biology. They look for DNA, for specific amino acids, for the waste products of Earth like metabolism. They are brilliant tools, but they are searching for a reflection of ourselves in alien mirrors. If a shadow biosphere exists here, it proves that life can be built from entirely different blueprints. This means our current approach to exploring other worlds is fundamentally incomplete. We are like explorers who only have a map of Europe trying to navigate the entire globe. We might land on the shores of a new continent and, finding no familiar landmarks, declare it barren, completely missing the civilizations thriving just beyond the beach. The discovery of a second Genesis would force a revolution in how we design missions. We would need to shift from looking for life as we know it to looking for life as we do not know it. This means developing entirely new concepts for biosignatures. Instead of searching only for DNA, we would design sensors that can detect chemical imbalances, patterns of complexity that cannot be explained by geology alone. We would look for strange ratios of elements in the soil of Mars, searching for a chemistry that defies the expected. On icy moons like Europa or Enceladus, we would not just test for our kind of organic molecules, but for any complex molecular system that seems out of place, any structure that shows a persistent, organized complexity. The goal would be to build a universal life detector, one that is not biased toward our own biology. This new perspective would even change how we study exoplanets light years away. Right now, we search for planets in the habitable zone, the Goldilocks region where liquid water can exist. But if a shadow biosphere teaches us anything, it is that life might not need water at all. Our list of promising worlds would explode. We would start looking at frozen moons with methane lakes, at scorching planets with ammonia clouds, at worlds we currently write off as hellish. We would analyze their atmospheres for any combination of gases that seem improbably stable, a chemical signature that hints at a hidden metabolism maintaining the balance. The discovery of a second Genesis on Earth would be the ultimate key, unlocking a much wider, weirder, and more wonderful universe. It would tell us that we are not alone, not because we have found another voice, but because we have finally learned how many different songs the cosmos can sing. This discovery would ripple far beyond the laboratory, shaking the very foundations of how we see ourselves. If a shadow biosphere is real, then humanity is not the sole inheritor of Earth. We share our home with a biological stranger, a neighbor whose family tree has entirely different roots. This forces a profound philosophical shift, a move away from anthropocentrism, the idea that humans are the central fact of the universe. We would have to embrace a true biocentrism, a world view where life itself is the central fact, and we are merely one of its many possible expressions. Our long-held sense of biological solitude would vanish. We are not alone in the universe. We were never even alone on our own planet. This realization could either diminish our sense of uniqueness or vastly expand our sense of wonder. Does a second Genesis make our own existence less special, or does it reveal a cosmos that is far more creative and prolific than we ever dared to dream? It suggests that the universe is not just capable of life, it is predisposed to it, that biology is a natural and likely outcome of cosmic chemistry. Our planet would become a living library containing at least 2 entirely different volumes of the Book of Life. This raises an even deeper question about the nature of reality itself. If 2 completely independent forms of life can coexist on one world, does that imply the possibility of parallel biological realities? Our reality is built on a left-handed chemistry of amino acids and a DNA-based code. The shadow biosphere would inhabit a parallel reality, operating on a different set of rules, literally occupying the same space as us while remaining chemically invisible. It is a staggering thought that multiple ways of being alive could be layered on top of each other, like radio stations broadcasting on different frequencies, unaware of each other's existence. This directly challenges our understanding of consciousness and intelligence. Our brand of life led to us to creatures who can look up at the stars and ask these very questions. But what other paths might a second Genesis have taken? Could a slow, silicon-based metabolism operating over millennia give rise to a different form of awareness, a consciousness so alien we would not even recognize it as thought? The existence of a shadow biosphere would not just fill a gap in our scientific knowledge, it would open a door to a universe of infinite biological possibility, forcing us to reconsider everything we thought we knew about life, our place in it, and the very nature of a living cosmos. Our relentless search for life might be the very thing destroying the evidence we seek. Every time a scientist collects a sample from a deep-sea vent or a desert rock, they subject it to a series of procedures designed to reveal our kind of biology. These standard protocols are a death sentence for any potential shadow life. The moment a core sample is pulled from an anoxic, high-pressure environment and exposed to our oxygen-rich air, it could be causing a catastrophic extinction event for a fragile, alternative organism. To our kind of life, oxygen is a vital gas. To a life form that evolved in its absence, it is a corrosive poison. Our sterile laboratory tools are cleaned with ethanol or bleach, powerful biocides that obliterate microbes. But what if these chemicals are not just sterilizing the sample, but actively dissolving the very cells we are hoping to find? A shadow microbe with a membrane made of crystalline silica or a gel of exotic lipids might simply disintegrate upon contact with our standard reagents, leaving behind no trace we would recognize as biological. Even our attempts to be helpful can be fatal. The nutrient broths we use to culture microbes are a gourmet meal for our bacteria, but to an organism with mirror image chemistry or a silicon-based metabolism, that same broth is a toxic sludge or a nutritionally barren void. We pour this liquid onto a sample, hoping for growth, and we might be watching a shadow organism starve to death or be chemically dismantled before our eyes, all while we note the sample as sterile. This is the contamination conundrum in its most tragic form. We may have already discovered and inadvertently destroyed our hidden neighbor simply because we did not know how to look after it. To have any hope of success, the search must become an exercise in extreme environmental simulation. It would require the creation of specialized laboratories that are the opposite of our clean, bright, and airy spaces. These labs would need to be sealed chambers where every variable is controlled. The atmosphere could be a pure mix of hydrogen sulfide and carbon dioxide, mimicking a deep hydrothermal vent. The temperature could be held at a steady 110 degrees Celsius, with pressures hundreds of times greater than at the surface. All nutrients introduced into this space would have to be meticulously crafted from scratch, built from mirror image amino acids or offering a diet of pure elemental silicon. Every piece of equipment, every glove that reaches into the chamber would need to be sterilized not with our poisons, but with their poisons, perhaps using a blast of pure oxygen or a rinse of pure water, substances that would be lethal to their kind of life. This is the profound shift in thinking required. We must stop trying to bring shadow life into our world and instead learn how to enter theirs. The true value of a shadow biosphere might not be in its existence but in its instructions. Unlocking a second biochemistry would hand humanity an entirely new technological toolkit, one designed by billions of years of alien evolution on our own planet. Our industrial world runs on chemistry that is often inefficient, energy-intensive, and polluting. We rely on extreme heat and pressure to manufacture plastics and fertilizers, processes that consume vast amounts of power. But what if we could borrow a better way? Shadow life, honed in environments we consider hostile, has already solved these problems. An organism that metabolizes sulfur at boiling temperatures possesses enzymes, the biological catalysts that perform miracles of chemistry with ease. We could harness these enzymes to create industrial processes that run at room temperature, slashing energy costs and eliminating toxic waste. Imagine a factory that produces vital chemicals not with roaring furnaces, but in silent, cool vats teeming with microbes that treat our pollution as their breakfast. The energy sector itself could be transformed. Our life is powered by the sun, either directly through plants or indirectly through fossil fuels, which are just ancient sunlight stored as carbon. But a shadow biosphere might run on a completely different power grid. Microbes deep underground derive their energy from the slow reaction between hydrogen and carbon dioxide, producing methane. What if we discovered a shadow organism that performs this conversion with 100% efficiency? We could design bioreactors that pull carbon dioxide directly from the air and transform it into a clean, limitless fuel, turning the tide on climate change with a biology that thrives on the very gases we are trying to eliminate. The potential for new materials is even more profound. Our biology builds with a limited set of components, primarily carbon, hydrogen, and oxygen. But a silicon using shadow life would have mastered the art of building with rock. It might produce biological silica structures with a strength and flexibility that our best material science cannot replicate. We could learn to grow electronics, not manufacture them, cultivating crystalline circuits that self-assemble at the nanoscale. Or consider a mirror life form whose right-handed proteins are indigestible to all known life. Those proteins could form the basis of a new class of super-durable, biologically inert materials, perfect for medical implants that the body would never reject or for coatings that never corrode or decay. The pharmaceutical revolution would be immediate and staggering. For decades, we have scoured the natural world for new drugs, discovering antibiotics from fungi and painkillers from poppies. But we have only been searching one shelf in the library. The chemical arsenal of a shadow biosphere would be a collection of compounds utterly unknown to our own biology. A mirror image antibiotic would be a magic bullet against drug-resistant bacteria. Our pathogens have evolved for eons to defend against our kind of chemistry. They would have no defenses, no resistance against a molecule that is the exact mirror opposite of anything they have ever encountered. It would be like fighting an army that has only ever prepared for an attack from the south, while we launch a full-scale invasion from the north. Cancer treatments could be redesigned from the ground up. Our therapies are often toxic because they struggle to distinguish between cancerous cells and healthy ones. But a shadow biology could offer targeting mechanisms of unimaginable precision. We could engineer therapies that use the unique chemical language of shadow life to seek out and label only diseased tissue, leaving everything else untouched. Perhaps the most futuristic application lies in information storage and computing. Our digital world is built on a binary code of ones and zeros, but life is the master of dense, stable, long-term data storage. A single gram of DNA can hold all the data of the entire internet. A shadow biosphere would offer a completely different model for a genetic code. What if its alternative genetic polymer is even more stable than DNA, or can store information in 3 dimensions instead of 2? We could learn to build biological computers that process information not with electricity, but with chemistry, operating in a fraction of the space and using a fraction of the energy. This would not be merely an improvement on our current technology. It would be the birth of an entirely new branch of biotechnology, a field built not on manipulating our own life, but on forming a partnership with a life we never knew was there. The economic and industrial potential is not just about new products, it is about new principles. It is about learning the rules of a game we did not even know was being played, and in doing so, winning a future we can barely conceive of today. This immense complexity is where human analysis hits its limit, but a new partner is entering the search, one capable of seeing patterns in the chaos. Artificial intelligence is now sifting through the microbial dark matter, scanning for the faintest glimmer of a biology we have never known. Machine learning algorithms can digest the entire global library of genetic sequences in a way no single researcher ever could. They are not looking for what is there, but for what should not be there. And our eye might flag a tiny, enigmatic sequence of code because it does not resemble any gene from any organism in any database on Earth. It could identify a strange statistical blip in the elemental makeup of cells from a deep rock sample, a pocket where the ratio of carbon to silicon is just a little bit off, hinting at a structure built from something other than pure carbon. These are the needles in a haystack the size of a planet, and artificial intelligence is the only magnet powerful enough to find them. Beyond just finding anomalies, AI is learning to dream up entirely new forms of life. Researchers are now training models on the fundamental rules of chemistry and physics. They ask the AI to generate theoretical blueprints for stable genetic molecules that are not DNA, or to map out plausible metabolic pathways that could run on methane or arsenic. These are not random guesses, they are computationally derived hypotheses for what a second genesis could actually look like. The AI becomes a digital explorer, venturing into a chemical landscape we have never visited, and returning with maps of potential biological territories. These hypothetical shadow genomes then become a guide for the physical search. Scientists can take these AI-generated patterns and actively look for them in real-world metagenomic data. It is like having a sketch of a missing person and then scanning through billions of faces to find a match. This transforms the hunt from a blind groping in the dark to a targeted investigation. We are no longer just hoping to stumble upon something strange. We are using the predictive power of artificial intelligence to tell us where and what to look for, turning the search for our hidden biosphere into a conversation between human curiosity and machine intelligence. One of the most visible and puzzling clues might be painted on rocks in plain sight. Across the sun-baked deserts of the American Southwest, on the stark faces of cliffs in Australia, a mysterious dark coating gleams under the harsh sun. This is desert varnish, a paper-thin film, often black or deep reddish-brown, that coats ancient stones. Geologists have long known that this is not just a chemical stain. It is a complex structure, rich in clay and iron and manganese oxides, that builds up almost imperceptibly slowly over thousands of years. The puzzle lies in its formation. In these arid lands, manganese should be virtually insoluble and immobile, yet here it is, concentrated into a hard, lustrous patina. Our known biology offers a partial answer. Scientists have found that certain bacteria and fungi can interact with the manganese and iron on rock surfaces, and they likely play a role in this process. But the full story remains frustratingly incomplete. The known microbes are present, but the sheer scale and consistency of the varnish and the precise way it accretes leaves room for a more profound mystery. What if the standard microbes are just the visible actors in a much deeper play? This is where the Shadow Biosphere hypothesis casts a long, intriguing shadow. Some researchers speculate that desert varnish could be the work of a hidden microbial community, a consortium of organisms with an alternative, incredibly slow metabolism. These would not be the bacteria we can easily culture, but something far more alien, living their lives on a geological timescale. Their energy source might be utterly mundane to them, perhaps drawing trace amounts of energy directly from the rock itself or from daily temperature fluctuations we would consider insignificant. Their biochemistry could be so slow and efficient that a single metabolic cycle, the equivalent of one breath, could take centuries. To such life, a rock face is not a barren surface. It is a thriving metropolis, and the varnish is the slow, deliberate construction of their city, a biological artifact built over millennia. This idea transforms desert varnish from a geological curiosity into a potential biosignature. If this is the work of a shadow biosphere, then the varnish is not just a coating, it is a living reef, the visible testament of a biology we have yet to comprehend. The evidence is not yet there to confirm this, and most scientists lean toward explanations involving our own known extremophiles. Yet the varnish persists as a beautiful enigma, a dark mirror on the landscape that reminds us that some of the best hiding places are not underground, but in the open, waiting for us to learn how to see them. The profound silence of the cosmos is the great unsettling mystery of our time. We gaze at a universe filled with billions of galaxies, each with billions of stars, and the question echoes, where is everybody? This is the Fermi paradox, the stark contradiction between the high probability of alien civilizations and our complete lack of evidence for them. But our search for a shadow biosphere here on Earth offers a radical and humbling solution. Perhaps we are not alone, we are just deaf to the conversation. The problem may not be that the universe is empty, but that our definition of life is far too narrow. We have been scanning the skies for radio signals, looking for planets with oxygen-rich atmospheres, searching for reflections of our own carbon-based, water-drinking biology. What if intelligent life is common, but it is built from a foundation we cannot perceive? Imagine a civilization that did not evolve on a planet's surface, but in the lightless, crushing depths of its crust, drawing sustenance from geothermal heat and the slow decay of rocks. Their entire existence, their technology, their very perception of reality would be built around sonar and pressure and seismic vibrations. A radio signal from the surface would be as meaningless to them as the faint crackling of static. Or consider a form of intelligence that arose not in water, but in the frigid methane lakes of a distant moon. Its thought processes, its communication, would operate on timescales we could never match. A single idea, a single sentence might take years to formulate and transmit. Our frantic gigahertz speed broadcasts would flash by in an instant, completely unnoticed, like a mayfly living its entire life in the space between two heartbeats of a giant. And then there is the most mind-bending possibility of all. What if such a civilization is not out there at all, but right here, sharing our planet in a form so alien we do not recognize it as life, let alone intelligence? We have considered the possibility of a shadow biosphere of microbes, but what if that hidden biology has, in its own way, achieved a form of sentience? A vast, distributed mind could exist within a subterranean network of silicon-based organisms, thinking thoughts of geology and chemistry over the span of millennia. Its art might be the slow, beautiful crystallization of minerals, its music the deep, resonant hum of tectonic plates. We would walk over its masterpiece cities, carved into the bedrock beneath our feet, and call it nothing more than an interesting rock formation. This reframes the entire Fermi paradox. The question is no longer, where are they? It becomes, would we even know if they were here? Our failure to make contact may not be a measure of the universe's emptiness, but a measure of our own blindness. We are like people who speak only one language, listening to the global cacophony and hearing only noise, completely unaware that the air is thick with 1,000 other conversations, all happening in tongues we have not yet learned to hear. The moment we confirm a shadow biosphere, we step into an ethical minefield unlike any humanity has ever faced. The immediate and most profound question is one of rights. What moral standing does a life form hold when it is not our relative, but our neighbor from a completely separate creation? Is it a resource, a collection of biological machines to be dissected and engineered for profit? Or is it a form of natural heritage, a priceless and irreplaceable testament to the universe's creativity, deserving of protection and reverence? Our history with newly discovered life is not encouraging. We have a long pattern of exploitation, of seeing the unknown as something to be conquered and consumed. But this would be different. This would not be a new species to add to our ledger, but a new kingdom of life, a second Genesis. To treat it as mere biological ore to be strip-mined would be an act of cosmic vandalism. The very discovery would force us to draft a new chapter in ethics, one that extends rights and considerations to a form of being that shares our world but not our ancestry. The risks of carelessness are immense and run in both directions. The greatest danger might not be a shadow pathogen wiping us out, but our own biology acting as an unstoppable plague against them. Imagine a researcher having successfully cultured a delicate, arsenic-based organism, accidentally exposing it to a common, harmless bacterium from our world. To that shadow life, our microbe could be an invasive monster, a biological bulldozer that unravels its fragile chemistry. We could inadvertently commit genocide against an entire domain of life before we even understand its basic principles. The reverse is also a terrifying unknown. While a mirror image virus might be biochemically invisible to us, what if its presence triggers a catastrophic, autoimmune-like response in our bodies or disrupts the fundamental processes of our ecosystems? We would be navigating a biological dark continent without a map, where the first misstep could have consequences we cannot even calculate. This demands a new global paradigm, a set of international protocols as stringent as those for handling extraterrestrial samples. Laboratories studying shadow life would need to be planetary quarantine zones, not just to protect us, but to protect them from us. The discovery would ultimately be a test of our maturity as a species. It would ask us if we are wise enough to be stewards not just of our own tree of life, but of an entirely different forest we never knew was growing beside it. The silence of the cosmos might not be a sign of emptiness, but a monument to a terrifying biological law. This brings us to one of the most unsettling answers to the Fermi paradox, the Great Filter hypothesis. It suggests that somewhere on the path from simple life to a spacefaring civilization, there exists a developmental stage so difficult to overcome that it effectively eliminates nearly every species that reaches it. This barrier could be technological self-destruction, a runaway AI, or something far more ancient and fundamental. What if the filter is not a technological trap, but a biological one already present here on Earth? Imagine a universe where second geneses are not rare but common. On countless young worlds, life sparks into being in multiple forms, just as it may have here. But in this cosmic struggle for existence, one form of biology consistently proves to be superior, not through intelligence, but through sheer, ruthless efficiency. This dominant biology could be a shadow biosphere, a form of life built on a more stable, more aggressive, or more versatile biochemistry. It would spread across a planet not as a predator we could see, but as a silent chemical tide. It would not compete for the same resources as carbon-based life. It would simply render those resources unusable or alter the planet's chemistry to make it utterly toxic for its rivals. In this dark scenario, our own existence is a cosmic fluke. We are not the pinnacle of evolution on Earth, we are the survivors. The shadow biosphere sharing our planet is not a benign neighbor, but the dormant, remnant population of that great filter. It is the evidence that the cosmic war for biological dominance was fought on this world long ago, and our lineage, by sheer luck or by finding a unique ecological niche, somehow slipped through. Perhaps it arrived on a meteorite, a biological weapon that sterilized a once thriving Martian biosphere before hitting Earth, only to find our planet's conditions temporarily inhospitable to its full, devastating potential. Or perhaps it is biding its time, waiting for a planetary shift or a trigger we cannot foresee. This transforms our search from a purely scientific quest into a matter of existential security. Finding a second Genesis would be the most profound discovery in history, but it could also be the ultimate warning. It would tell us that the universe is not a safe garden for life like us, but a battlefield where most life is quietly, systematically, and permanently eliminated. We would have to look at every new exoplanet we discover not with hope, but with dread, wondering if the great filter is already there, waiting. We need to clear away the science fiction to see the true science. The shadow biosphere is not a realm of mythical beasts or spectral entities. You will not find Bigfoot lumbering through these hidden forests, nor ghosts drifting through its silent cities. This search targets something far stranger and more profound, the realm of the microscopic. The inhabitants of this potential world are not creatures of legend, but of chemistry, single-celled organisms so alien in their fundamental construction that they have remained invisible to our most advanced tools. They are not hiding in the remote wilderness, but likely in the soil beneath our feet, in the rocks deep below, or floating beside our own bacteria in a drop of water, completely unnoticed. Another common mix-up involves the very origin of this life. This is not the concept of panspermia, the idea that life was seeded on Earth from another world. A meteorite carrying Martian microbes would be an incredible discovery, but those microbes would still be part of a single cosmic tree of life. They would share our basic biochemical framework. The Shadow Biosphere hypothesis is far more radical. It proposes a second genesis, a completely independent origin of life that happened right here on our own planet. This life would not be our long-lost cousin from space. It would be an absolute stranger with no relation to us whatsoever, a true biological neighbor that originated in its own primordial soup. And do not expect this hidden life to be complex. After billions of years, our own tree of life produced redwood trees, blue whales, and human beings. A shadow biosphere, if it exists, almost certainly never progressed beyond the microbial stage. Its organisms would be masters of survival, not complexity, persisting in a cryptic state for eons. They are not building civilizations or using tools. They are simply existing, carrying out their slow, strange metabolisms in isolation. This is why they have remained hidden for so long. They are not intelligent beings playing a cosmic game of hide and seek. They are simply following a different set of chemical rules, living in a parallel biological reality that has until now been just beyond the edge of our perception. The hunt is no longer theoretical. Across the globe, a new generation of scientific missions is actively probing the hidden corners of our planet, armed with tools designed to find life as we do not know it. One of the most ambitious of these is the Deep Carbon Observatory, a decade-long international project that has acted as a census for the subterranean world. Its researchers have drilled kilometers into the seafloor and sampled rocks from deep mines, revealing a startling truth. The subsurface holds a mass of carbon-based life rivaling all the life on the Earth's surface. But for those searching for a shadow biosphere, this vast, dark realm is the ultimate playground. The project is not just counting our own kind of microbes, it is analyzing the chemistry of these deep environments with unprecedented precision, looking for anomalies in energy flow and elemental ratios that could betray the presence of a hidden metabolism operating just beyond our detection. It is systematically mapping the darkness, searching for a light that does not match our known spectrum. Meanwhile, the lessons from the arsenic bacteria controversy have directly shaped research at NASA and other space agencies. The focus has dramatically shifted from hunting for familiar signs of life to developing agnostic biosignatures. Scientists are creating instruments that do not look for DNA, but for patterns of chemical complexity that cannot be explained by geology alone. They are building sensors that can detect the subtle-handedness of molecules, hoping to spot a pocket of right-handed amino acids amid the overwhelming left-handedness of our biosphere. On the drawing board are microscopes that use novel forms of spectroscopy to image the interior of individual cells, searching for structures that defy all known cellular architecture. This is not just about new hardware, it is about a new philosophy of detection. Private initiatives and interdisciplinary collaborations are pushing the boundaries even further. Teams of astrobiologists, synthetic chemists, and computer scientists are working together to design what they call a universal life detector. Their goal is to create a device that can be placed in any environment, from a Martian crater to a deep Earth borehole, and simply report back on whether it finds any form of organized, complex chemistry that is out of equilibrium with its surroundings. It would be a lie detector for the universe, capable of sensing the whisper of a metabolism regardless of its biochemical basis. These projects represent a fundamental admission that to find something truly new, we must first build the tools to see our own blind spots. The search for our hidden neighbor is now a tangible, funded, and global scientific endeavor. moving from the pages of speculation into the rigorous world of testable hypothesis and discovery. Yet the most profound result of this search might be a resounding silence. What if, after decades of meticulous investigation with the finest tools we can devise, we find absolutely nothing? The complete absence of evidence for a shadow biosphere would send a chilling and lonely message across the cosmos. It would suggest that the spark of life on Earth was not a common event, but a singular, miraculous accident. The leap from non-living chemistry to a self-replicating system would appear so astronomically difficult that it may have happened only once in 4.5 billion years on this entire planet. In this scenario, we are not just alone on Earth, we are likely alone everywhere. The great silence of the universe would not be a puzzle, but an answer, a confirmation that life of any kind is an exquisite fluke, and that our own existence is a breathtakingly rare cosmic performance with no other audience. This outcome would fundamentally reshape our self-perception, casting humanity not as one voice in a galactic chorus, but as a solitary whisper in an immense and empty hall. But science thrives on ambiguity, and a null result is never a simple dead end. The failure to find a second genesis could just as easily point to the limitations of our own perception. Our entire search is constrained by the very chemistry we are trying to transcend. We are building detectors for a signal whose frequency we can only guess at. It is entirely possible that a shadow biosphere does exist, but in a form so intrinsically cryptic that it is forever beyond our reach. Its organisms might not form discrete cells that we can isolate, but exist as diffuse, network-like structures integrated into the rock itself. Their metabolism could be so slow that it is indistinguishable from the background weathering of minerals, a biological process that takes 10,000 years to complete a single cycle. Or their signature might be collective, only becoming apparent across entire continents, a pattern we cannot see because we are standing too close. Our most advanced machines are still crude compared to the subtlety of nature. A failure to find shadow life now does not mean it is not there. It only means we have not yet learned how to look. The search itself is what pushes science forward. Every refined instrument, every novel experimental design, every clever algorithm developed to hunt for our hidden neighbor expands the very definition of what is detectable. We are learning to ask the universe better questions. Even if the ultimate answer remains elusive, the journey transforms our understanding of life's potential, teaching us to see the familiar world with alien eyes. The value lies not only in the destination, but in the profound expansion of our imagination along the way. Our journey has taken us from the deep oceans to the desert varnish, through the enigmatic world of microbial dark matter and the silent hunt using artificial intelligence. We have seen how the very tools of science, once a barrier, are now being redesigned to find what they once overlooked. The staggering implication remains that our planet could be a library with two entirely different volumes of life's story. Confirming a shadow biosphere would not just fill a gap in a textbook, it would fundamentally reorient our place in the cosmos, telling us that biology is not a lonely accident but a cosmic imperative. This quest is the ultimate lesson in scientific humility, a reminder that what we do not know is always vaster than what we do. It asks us to look at a handful of soil or a drop of pond water not as something familiar but as a frontier. The real discovery may not be a microbe in a lab, but a new way of seeing the world around us, to sense the profound and hidden possibilities teeming in plain sight. So we leave you with a single question to ponder. What do you think? Is there another form of life sharing this planet with us? Let us know your thoughts in the comments and subscribe for more journeys to the edges of our understanding.